Good evening, everyone. Hello and welcome to the Stephen and Peter Sachs Museum. I'm Neshka Pfeiffer, the Sachs Museum Curator, and your host and moderator for the Grafting the Grape online program series, which is sponsored in partnership with the Missouri Humanities Council and with support from the National Endowment for the Humanities. Today's presentation, Art and Science Dialogue, is a moderated discussion with contemporary artist Dornith Doherty and botanist Dr. Allison Miller, and we'll begin shortly. This is the 5th presentation in the Grafting the Grape series, and we're very excited to have you join us. There are 6 presentations in this series, and they highlight content from the current exhibition at the Sachs Museum, Grafting the Grape, American Grapevine Rootstock in Missouri and the World, which is now open to the public at the Garden. The previous event's recording is now available, and I'll drop a link in the chat for anyone who's interested in that talk. We are able to offer accessibility features, including ASL interpretation for the deaf and hard of hearing, and live captioning, which you can access via the closed caption button at the bottom of your Zoom screen. To turn off or for more options, click on that closed captioning feature. A note about today's webinar: it will run approximately 1 hour. Our dialogue will last about 40 minutes, with time at the end for a Q&A session. Throughout the webinar, we encourage you to use the Q&A function in the box at the bottom of your screen, and we'll sort through your questions and get to as many of them as possible at the end of the presentation. Today's program will be recorded and posted on the Missouri Botanical Gardens YouTube channel in the coming weeks. We're joining you from the Stephen and Peter Sachs Museum, which I'd like to acknowledge is on the ancestral and occupied land of the Chickasaw Nation, Illini Tribe, Iowa Tribe, Kickapoo Tribe, Osage Nation, Oto-Missouri Tribe, and the Quapaw Nation. We acknowledge the ongoing relationships these nations have with this land and urge non-native people to educate themselves about this history and about the contemporary work of tribal sovereignty within these nations. Through this acknowledgement, we honor the elders and their descendants of these indigenous nations in order to bring visibility to these long and silent histories while working toward a more just and equitable future. Our speakers today will discuss the impact of the grape phylloxera epidemic on European vineyards that led to the development of using Native American grapes for grafting, and how this history and science intersects with the artwork series created for the Grafting the Grape exhibition. Dornith Doherty is an American artist whose work is concerned with human entanglement in the rapidly changing environment. A 2012 Guggenheim Foundation Fellow, Doherty has also received grants from the Fulbright Foundation, the Japan Foundation, and the United States Department of the Interior. Working primarily with photography, video, and scientific imaging, Doherty's work is in numerous permanent collections and has been featured in exhibitions and publications widely in the US and abroad. She is Distinguished Research Professor at the University of North Texas, where she's been on the faculty since 1996. Dr. Allison Miller is a professor of biology at St. Louis University and member and principal investigator at the Danforth Plant Science Center, who is also the lead for the Vitis Underground Project, which is a 5-year multi-institution project investigating the relationship between root, scion, and environment in grafted grapevines. The results of the Vitis Underground study will facilitate the development of perennial crops with enhanced phenotypic robustness and plasticity, enabling them to thrive in a range of climates. Welcome, Dornith and Allison. Thank you so much for being here this evening. We're so glad to have you. And since I'm off screen, I get to be the master questioner, right, to kick off this dialogue. And I'd like, Allison, maybe for you to start with, to tell us a little bit about how you came to research American grapevines and their rootstocks, to sort of tell us a little bit about why grapes are so important.
Sure. Well, as Neshka mentioned, I'm a botanist, and the way I came to North American grapevines was understanding the diversity of grapevines that are here in Missouri. There are 8 or 9 native species in Missouri and 19 native grapevines in North America. There are another 37 in Asia. So we came to know these species in the forests of the surrounding areas and started thinking about their natural diversity and population structure. And it was in the course of that work that we came to learn of the role of these grapevines in global viticulture. And it was in the mid-1800s that phylloxera, this aphid that we'll hear a lot about, was introduced from North America into Europe. And ultimately, the Native American grapevines that have inherent resistance to this phylloxera, this bug, this insect that basically chews the root systems of grapevines, the North American vines can withstand it, but the European vines cannot. And that is how grafting kind of got started. And our interest in the current research project was trying to understand what else those native grapevines do as in their role as rootstocks. So once a European vine is surgically attached to a North American grapevine and its roots are then native to North America, how does that change the rest of the plant? And that's really the core of our Vitis Underground project, is trying to understand how different rootstocks impact the leaves, the physiology, the patterns of gene expression, the chemistry of the berries, and even the volatiles that the wine that the wines carry as products of these grafted vines. And what we're finding is that it's a really complex series of relationships that vary based on the genetic entity or identity of the rootstock, based on the scion that it's attached to, and based on the location where that vine is growing. It's very dynamic and really quite exciting, actually, to think about how these sedentary organisms sense and respond to their environments over time and the role that the roots play in that.
You always put that so beautifully and so concisely.
Oh,
There's a lot of science in there.
And I love that title, Vitis Underground. That's genius.
Well, that was one of our collaborators came up with that very early on, and it has stuck, and we really enjoy it. So it describes the work.
So are you using all of the native grapevines or just a selected few in your studies?
Well, so yeah, great question. There are, as I mentioned, 19 native grapevines in Missouri, but there are only 3 right now that play the primary role as rootstocks, although there's some really interesting work happening in California and elsewhere with some other species that historically haven't been essentially used as rootstocks, but might play an increasingly important role, particularly as our climate dries and warms in some parts of the grape growing areas. So yeah, so the species that are used, I don't know if this was your next question, Dornith, but the species that are used for rootstocks are Vitis riparia, Vitis rupestris, and Vitis berlandieri, and it's most often a hybrid derivative, so the product of a cross between 2 of those species that are used as rootstocks.
And so what drew you to this project when I reached out to you, actually on Allison's recommendation, to create work for this exhibition?
Well, as you mentioned in my intro, that I have a huge interest in stewardship of both the land and the ecosystems that are part of that land, and kind of sustainable agriculture, but also preserving biodiversity in the wild world. I really have a big interest in that. And, I had met Allison at the Prairie Festival, where she presented her research on her work on sustaining grain agriculture through,
They're these perennial herbaceous species. And the Land Institute that hosts the Prairie Festival works primarily with Kernza. Mm-hmm. That might be the one, and that's the trade name for intermediate wheatgrass, or Thinopyrum intermedium, Silphium integrifolium, and several other species.
Right. And so I was super impressed by your work and just the kind of what the Land Institute is doing towards perennials and preserving, wild perennials, and by making them more attractive to human use. So as a strategy of doing that, like everybody benefits through that. So I was super interested in Allison's research, and then just the opportunity to work with her and her lab. And also the same day I got the email from you, I also got one from Keith Duncan talking about the X-ray machine at the Danforth. And I was just, that it's huge, it's a CT scanner, you can stand in it. He was like, come on down, I've seen your work, with the Seed Bank project with Archiving Eden, and I'd like to, see if you'd be interested. And so that combination, the confluence of all that, I did a research trip and met with you, had a chance to tour and, see the beautiful facility that is the Missouri Botanical Garden and the herbarium, and then see your lab and the Danforth. It was just such a great, so there were a lot of toys and a lot of interest, but also just the whole importance of this cycle. And of course, I had become very interested in the Columbian exchange through my work on global seed banks. So for those of you who don't know what that is, that's the kind of exchange of ideas and infectious diseases and plants and human slaves that marked that exchange between when the Western Hemisphere and the Eastern Hemisphere were connected, usually dated around 1492. So when I heard, started researching the Phloxera, the idea that this exchange, that it was part of the Columbian exchange, but that, that it started going right at the very beginning, but because of technological advancements, i.e. at that point, the steamship, they could go faster, right? And then they started surviving. So I was fascinated by that. I also have a big interest in, as I've worked for the last dozen years on Archiving Eden, which is still ongoing, I became very, very interested in different kinds of plants and how they tie into everyday, current research, but also their past. So one of the things that I'd been interested in was potato blight, and there's a lot of similarities between how the potato blight happened and the phylloxera epidemic. So, so that was super, super interesting to me from that it's an important, we're super dependent on it. It's not quite like potatoes, of course, where people died, but still, people were very economically dependent, and of course, wine is fantastic.
So what we're going to do for the audience is we're actually going to, Dornith created 3 sets of works that's a part of the entire series that she created for the Grafting the Grape exhibition. So the first thing we're going to do is we're actually going to look at her Phloxera images, 2 of which you can of course see behind both Allison and Dornith, but we are going to get a little bit more detail. So these are what I'm going to do is just briefly thumb through them so everybody can see all 6 images. And Dornith, maybe you can explain a little bit about your process how these images were created.
So Phylloxera, the insect itself, is tiny, 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 and was not visible. That people didn't really tie it together because they hadn't really seen the insect. And correct me, of course, because you know a lot more about this than I do. So one of the things that I've been in my other work, I'm interested in how making the invisible visible through either scientific technology or photography, but capturing images of things that are invisible to the human eye that are otherwise invisible, that you can't see unless you use some aid. And one of the first things I wanted to do was just look at what does a phylloxera look like. Then Nesca, as we were working, you of course were trying to source it and posted on a listserv for entomologists and museum professionals where they were combating pests. You know this story? It's a great story. So she posted and said, I need to actually, instead of combat a pest, I need to invite one in. And the only person that responded was an entomologist in Australia. And so as part of this kind of total Columbian exchange thing, these phylloxera are from Australia. And he had some just growing in his backyard garden. And he prepared them so that I did not, release phylloxera into our clean room at the university. And so I was expecting the insects, and what I received, I was so happy to see this, are these very beautiful galls. And the galls are created when the female phylloxera covers herself in her own eggs, and then they hatch. So these are these architectural, really beautifully patterned, almost mandala-like structures are actually the egg casings for phylloxera. And what's amazing is it speaks to the fertility of this pest, but also the biodiversity of it, that all of these, even though they're different, are also the reflection of an individual plant, insect's life that has been expressed in this very particular way, so that they're very, very different. So that biodiversity and then the fertility of that comes from biodiversity, I thought was super, super interesting. They're printed they're gold toned, and part of that is for a couple of reasons. One is that when you're capturing an image with an organic matter with a scanning electron microscope, which is how I made these, many times you have to coat it, you have to treat it so that it does not, so that you can capture a good image. And most of the time they do a non-reactive surface gold. I did not do that. I had been experimenting with other dry organics as a way of imaging, so I was able to make this without that. But they reflect the time period of the mid-1900s when photography was invented, when the phylloxera came. These appear, if you see them, get to see them in person, they look like orotones, which are images that are on gold, that are photographic.
And Allison, how does phylloxera impact your work today?
Well, it's interesting, as you were talking, I was thinking, Australia, we have phylloxera all over our vines, but I think the timing of when they were needed, it was not the right season here. So phylloxera, the main problem for viticulture, for growing grapevines with phylloxera, is the stage of the aphid's life cycle, which takes place below ground. And there is a below ground part of its life, and that's the part where it sucks the sap out of the roots and causes these problems for species that haven't evolved the capacity to withstand it. The part of the life cycle that you've captured, I think, is the part that is above ground on the leaves. And this is, so after they kind of live underground for a while, they emerge and generate these galls on the leaves. And so how it affects our work today is that many of our experimental vines, by the end of the summer, this time of year, are covered in these galls, the leaves. And part of the work that we're doing is quantifying variation in leaf shape and other traits of the leaf, which can become very difficult because it's not often just one or two galls on a leaf. It's an entire village of these galls, and it actually causes the leaf to become deformed and to kind of roll up, and it's problematic. And we've actually looked to see how phylloxera is distributed across our vineyards, and it seems to be random. We have a lot of different genetic individuals within our experimental vineyards, and there don't seem to be particular individuals that are, at least in our experiments, that are more or less susceptible. It's just there'll be a patch of it that will pop up in one corner of the vineyard and spread from there. So practically it's challenging. It doesn't kill our vines because we're not working with straight European vines, but it does make for some interesting sampling in these experimental vineyards.
That's great, thank you. Do you have any questions for Allison about phylloxera?
I don't think so.
Okay, so what we're going to do next is move to your roundabout circuition film first. So bear with me, everyone, while I share the screen here. So would you explain a little bit, Dornith, what everyone's viewing, the clip with the...
That is a really brief clip of a longer piece that is meant to be pretty contemplative. It's just a 1-minute clip from like a 7-minute total video that's projected here in the gallery on the wall. And it is called Roundabout Circuition because of it's a metaphoric title for all the different cycles that are embedded in this story. There's the Columbian Exchange, there is the life cycle of the Phloxera, there is the life cycle of the grapevines as they battle Phloxera, and there's the seasonal variations, of course, that everybody knows from, spring to summer to winter to fall. I mean, to fall to, you know what I mean. I did that out of right. So, but anyway, so as you go through this, it is not an illustration of how Phloxera attacks or affects, but it does have that sense of there's a flourishing and then there is a decay, and the background is this very kind of stormy sky that changes color and over time, that talks about, the kind of the threat part of it. So that's what that was. And it's an interesting, because it's like also has a pandemic aspect to it. I was originally going to come and take cuttings and samples from the actual vines that Allison was working on in these experimental vineyards, and there's a small one here at the Missouri Botanical Garden, I think that's still there, right? And I was unable to travel because of the pandemic. So fortunately, there's another part of the story of the Phloxera, which is the Texas aspect, that, when the grapevines they introduced Phloxera accidentally, and then they actually rescued them by sending more roots that were able to resist the Phloxera infestations. Part of that story came from the research of a guy in Texas as well, who also sent grapevines. So there are a lot of wild grapevine species in my area. In fact, my zip code has a town that's a historic town called Grapevine in it. And that is, so there's wild grapevines, there's actually a vineyard there. So I was able to source a couple of different varieties using the descriptions that your grad students told me about. Right? They were like, well, you had talked about the riparian one, that there's one that's ground-based, there's a climber, and I was able to kind of identify, plus they have very different leaves. So I was able to source a bunch of different leaves and vine material and grapes and grapevines through just, foraging, and also foraging in this vineyard. So that is how I ended up doing the workaround for the pandemic, is that those are actually actual wild grapevines combined with one vineyard that had a really bad case of phylloxera when I was there.
So for Allison, when you're testing and working with grapevine plants, what are their most unique characteristics? Like, what makes them stand out? Like, do they present special difficulties to study?
I think, Dornith, actually you hit on it as you were describing your exhibit here. What draws me to grapevines is their perennial nature, and that is the major focus of my research, is on perennial or long-lived plants. And among long-lived crops, and almost all perennial crops are woody, so they're either trees or vines. Grapevines are really a terrific model because we know a lot about them, because wine is hugely economically and culturally and socially important, and there's excellent funding opportunities for research in grapes. So what draws me, the kind of the bigger question, is how does a long-lived organism persist through the course of seasons, from one year to the next, and over the course of decades, and in some cases centuries. And in perennial crops like grapevine, about 75% of these crops are clonally propagated, which means that they're genetically identical to one another. And they're different varieties, each of which is clonally propagated, but they're really genetically quite homogeneous. And so then when you think about how these very genetically similar organisms are sensing and responding to their changing environments. And, plants can't move. These guys aren't going anywhere. They're stuck wherever they get planted. So it's really quite fascinating. This is a phenomenon that we call phenotypic plasticity. It is the range of different traits or phenotypes that a single genetic individual can display. And so grapevines are a great model for trying to understand that. And as the climate is changing, we can look at California as an example right now. Those vines, the vines that are there today, are in most cases are the same vines that were there 10 years ago or 20 years ago, and how they're dealing with the smoke and the heat. It's just really interesting to think about that. And the rootstock part of it is grafting. That process kind of decouples the process of development or breeding of plants. So the grafting, breeders are selecting for root systems that have the effects that they want on the shoot systems, the above ground part of the plant. So part of this project has been not only understanding how the vines change over a season and from one year to the next, but also how the root systems can either constrain those changes or allow them to exhibit a wider range of phenotypes. So it really is the change over time of single genetic individuals that is, I think, so fascinating. And I'll just note, it's not just crops. The majority of the world is perennial and from a plant diversity perspective. So it is quite interesting to think about how this work might apply beyond the grapevines.
And when we talk about climate change, because we know some of the things you've mentioned, have you been able to identify specific stressors on the plants that make them adapt, that are measurable in the time that you have the ability to look at them?
Unfortunately, yes. We've seen a lot of problems. Like something that we see a lot in Missouri is we'll have a warm spring and the vines will think, oh, great, it's going to be summer and they put the leaves out and the vine starts going, the flowers come out, and then there's a late spring frost. And we've seen this the last 2 summers, or 2 springs here, where our experiments were decimated because the vines are perceiving this warmth early, and then they get kind of shut down pretty severely, and then have to come back from that. And it's not that this has never happened before, it's just that we're seeing it seems to be increasingly severe and common, I guess, this at least here, this late spring frost. Early warming, late spring frost.
In your roundabout film that we saw a glimpse of also, Dornith, you were referencing part of the narrative. Your narrative is also including climate change. Is that true?
Yes, absolutely. And part of that is not, again, I'm not trying to illustrate it, but reference it through these different cycles and the kind of struggle that, there's a period of the video, well, it's going on behind you now, where it's just, completely wiped out. And then there's the little bit of hope where the regeneration part of it. So that, and then of course referencing the sky in the background with the climate change.
And I'm going to just slightly shift a little bit here, because both of you have talked about, like Dornith, you've done work on the potato, which of course had a big blight, which we were also quite lucky to show here in a previous exhibition on the potato. And Allison, you mentioned the fact that grapevines are, at least the grapevine berry, are clonally propagated, and we know that was also the challenge with the potato in the 19th century too. So are there other crops right now that are also clonally propagated that we have to be concerned about, especially for the marketplace, but in terms of future genetic diversity and looking at older and other plants?
Yes, oranges are an example, bananas are an example. There's a lot of, as I was saying earlier, about 75% of long-lived crops are clonally propagated. But this kind of gets back to a point that you brought up in describing your Archiving Eden project, which is what I gather or took from that exhibit is it's just a way to highlight the variation that's inherent in different species and how many different species are out there. So one of the things that is a major focus of research here and elsewhere is genetic variation that occurs in nature that could be potentially valuable as we try to adapt crops for these changing conditions. And so on the one hand, grapevines, single vines have to respond, as they can, as wherever they are through their lifetimes. But if we think about kind of trying to breed or adapt to those crops, we can do it by crossing, one way is by crossing crops with their wild relatives, which is why we're interested. It's how we actually first got interested in studying natural variation in the genus Vitis here and elsewhere. So I see a lot of hope actually, because there's a tremendous amount of natural variation that is relevant for agriculture, that is being studied and preserved, but I think still there's a lot out there yet to discover and a lot that we can do to hold on to that variation and potentially use it as we begin to think about what agriculture will look like in the future. And places like the Missouri Botanical Garden, I think, are absolutely critical in knowing and conserving that natural variation, well, for agriculture, but for everything.
Like you, I'm totally on the same page in terms of preserving as much biodiversity, whether it's a crop or not. Yep. Because, if you think about crops that have co-evolved with us, like corn, right? Where, it started out as a little grass with a tiny, super hard seed covering called teosinte, and then over the 2,000 years that it's been domesticated, that it's evolved into this just brilliant amount of diversity that's gone, that is through its relationship with humans, right? That it's traveled around the world. They're doing those dry land tests in northern Africa with corn, but then also, we're at risk of having teosinte go extinct in the wild. So trying to collect all those variations where they're on the fringes and they relate to the corn is super, super important. But if you use that as the model, right, where the corn in the US is, the agribusiness corn has a very, very narrow range of it's super much the same. But then there's all these other kind of corn. And so trying to save that as a model, because you don't know what a particular trait will do in the future, or what unknown pandemic will arise that the plants themselves will need, and they'll need that kind of genetic diversity to kind of, hopefully there'll be some range that will, in the way that we're trying to figure out why do some people get more sick from COVID, right? And there's got to be a genetic little tiny, you're the scientist, but so I won't put words in your mouth, but I'm assuming that kind of biodiversity among the human population is also helpful.
Yeah, and the only thing I would add to that is I think grapevines are a really terrific model for an example for why it's important to conserve variation in the wild, because the grafted grapevine was possible because there are these native species that are by themselves not used to make wine, but they harbor traits like resistance to phylloxera that turned out to be really important for the global grape crop. And in this case, it was introduced via grafting, which is another horticultural technique that's slightly different than the breeding that I was discussing before. But nonetheless, it was a wild species that nobody would have thought had any use, but actually was critical for the industry as it exists today. And so I think I think grapevines are really a nice way to emphasize and illustrate why the conservation of wild biodiversity is important for agriculture.
And it's interesting because it's like corn, it's co-evolved with us
Yep.
over you know, millennia.
Yep.
So the fact that there's still wild versions of it, you know, if you think about that, that's pretty interesting, right? That there's like so many of the specific named varieties, and then there's still all this wild that hasn't really been tapped. That's a really interesting part of it, right?
And that's the hopeful part of it, is we have not exhausted the possibilities, right? There's still a lot to be discovered.
And so what we're going to do next is look at the third set of the series, so the final set of artworks, which is another short film and this is also called circuition, and this is the 3D scans of the rootstocks.
Yes.
So please go ahead and tell us a little bit about what people are seeing.
So if y'all remember when Allison was introducing her work, she talked about having three main rootstocks that she was interested in. And so she very generously loaned examples of those three rootstocks. And then Keith Duncan at the Danforth Plant Institute made CT scans of them, which I was then able to use as a way of going through all the possible variations if you have three variables, of crossing between two with three variables. So, you know, it's just a sample of what is projected on the wall here, and the color is not real color. It's really to make it be so that you can identify the different rootstocks as they rotate through all the possible variations.
And had you worked with roots before? Had you ever included roots as a part of your artistic?
I have not, and well, I say that, and then I'm like, oh, think of all the different things. You know, they came up when I was doing X-rays with the seed bank work. I was doing X-rays of tissue samples, which are little plantlets that they have either from cloned or from seed tests and at seed banks around the world. So I have indeed made still images of them, but with the access with the collaboration with Allison and Keith allowed me to do a video of roots. And what's interesting about roots is, of course, that you can't see them, right? And that they're, again, invisible to, without some kind of aid to see them underground. There's this mystery of what's going on down there, all sorts of ways that people try to figure it out while still keeping them in the dirt. And that's what's so remarkable about these. If you look at them, they're all grapes, and yet each one of these three grapevines are just totally distinct. I mean, their roots are totally different. And it is, so you're seeing not only what's invisible because it's in dirt, but also the inside of the roots themselves.
So Allison, can you tell us why do you scan the rootstocks this way?
Well, honestly, originally this was not part of our research project, and Dornith mentioned Keith Duncan, who is a research scientist at the Danforth Center, and the lab that Keith works with is run by Chris Topp, and Chris is a leader in developing approaches for characterizing variation in roots, which is inherently difficult because you can't see it. But I mean, if you think about a plant, half of its body lives above ground in air, and the other half of it is below ground, living in soil, and we really have no way, historically we've had no way to see what that's all about. So the Danforth Center and the Top Lab has pioneered a number of really interesting, innovative, cutting-edge approaches for capturing root architecture and size and growth rates, and just amazing. And so we had a little experiment going in the greenhouse, really a side project, where we had the same scion or top part of the plant grafted to different rootstocks that we had ordered commercially, because we wanted to see if we could try to replicate in the greenhouse some of the traits that, or the features that we had picked up in our experimental vineyards in the field. And just in casual conversation, it just happened that Keith said, well, we could scan some of those. And we thought, oh, great. You know, so after this experiment, we actually scanned them before we potted them, because they had been, the way commercial vineyards make grafts is they grow them for a year and then pull them out of the soil and then clean them off and store them, and then the next year they go in the ground for real. So the vines that we had obtained for this project, Keith scanned them at the front end of the project, then we grew them in the greenhouse as part of an experiment, and then he scanned them at the end of the project so we could begin to capture some of the changes below ground. And again, as I mentioned, it was just kind of a hallway conversation that got going to more than we thought, and even more when Dornith came on the scene and was able to use that material to make these really incredible images. But we think a lot about what's going on below ground and are trying to incorporate that into our work, in part through collaboration with Chris and others. And it's just not, it's so important and it's so hard. So I think we'll see a lot of, and we're already seeing today a lot of interesting technological advances in this space. And so I feel fortunate to work in a place where people are doing this, because otherwise we'd be in the dark.
Well, and that's the interesting part about art-science collaborations too, right? Is the same experience that you had with Keith, where it's a hallway conversation and starts with what if, right? And that's what's been really, this show took so many different directions because of conversations, which is really, I love that part, right? That you hear about, like, oh, that's so cool, really? Or like, it's on the Edwards Plateau. It's like, well, you should come. You should see what that looks like, because it's beautiful. Yeah. So it's, that's really been a real pleasure to work with you and Neska and Keith, who's not here today. So.
Well, and from the scientific perspective, it's so interesting to see these structures through the eyes of an artist. And as art, because for us, I don't often think of plants in that way. And it just really changes, even thinking about phylloxera on the leaf, often our attitude is like, oh no, like there's more, how are we gonna deal with this? And it's not a thing of beauty always. But when I first came into this exhibit and saw these phylloxera images and the roots, it just changes my perspective. So I totally agree, and I think artists and scientists, I think we'll see more of this in the future, I hope.
Yeah. And Dornith, I know, like, you mentioned that when you are speaking with scientists, sometimes it brings something to the fore that you weren't expecting in the way that you proceed in your work.
Oh yeah, no, it's totally unforeseen, right? Like even this one came out of the idea of, like, Gregor Mendel's experiments with, what was it, sweet peas? Yeah, peas, right? About, A plus B, like how I learned statistics in junior high, right? And like the 1 week we spent on statistics had to do with Mendelssohn, right?
Yeah.
Gregor Mendel, not Mendelssohn, right? And so that part about, if you have 3 variables, like how many possible combinations can you do, really came out of your research. Right.
Yeah. Well, and the other benefit, I think scientists have a long way to go in how we communicate science to the public. And when I first saw your work at the Land Institute, what really caught me was I had a visceral reaction to seeing those seeds. And that's something that we often cannot elicit from people. It's with our tables and our principal components analyses and whatever it is that we're doing. And I think it's so important right now that we capture attention. And I believe more and more that art is a really important way to do it. And there's so much beauty in nature, but there's so much left to learn and left to discover. And maybe it's through art that we can help share that with people and help bring them in to, at least maybe not necessarily to be scientists, although that would be great, but just to appreciate science and the role of science.
Just have a moment of contemplation, right? That maybe ask the question of, like, how can I participate? How can the force of individual action. I mean, clearly climate change is a big thing, but individual actions add up, and really, like those discoveries that we're talking about are very small groups of people, right, are making really big differences. So for me, it's that, and it's interesting to me because when I started that science project, the seed bank project, all the scientists literally were like, in the generous spirit of scientific collaboration, sure, come on in. They were mindful that, you can't just, nobody can just walk in, but, they allowed me to collaborate, and their idea, but they were convinced. They were like, there's not a project here. There's nothing here that anybody wants to see, because what we do is important, but it's not visual. And I was like, well, let me just see what I can do. And then all of a sudden it's like this beauty. Like, who would ever think that the phylloxera would be quite as beautiful as this is?
Or roots.
Right, or roots. Those,
yeah. To be honest, it is, as I'm looking at it, it's really beautiful. But yeah, I think it's an important thing. And I like to see that scientific organizations and funders like the National Science Foundation are supporting these types of collaborations, because it's important. It's a really effective way to help convey what's going on in the scientific world, and it's an exciting world. So however we can get people to see it is,
or just ask the question, right? Like you don't really learn anything from this exhibition, but maybe it sparks a question in your mind and starts a conversation, which is what I'm interested in. It's like, you can get people to be curious about it and have a conversation. They may really, benefit.
Yeah, I totally agree.
So I think as we wrap up for the evening, I have 2 questions for you, one for each of you basically.
Okay.
So tell us maybe, Allison, a little bit where the Vitis Underground project is going to wrap up, or where it will be in the next year, right? And Dornith, what are some other projects what are some other plants you might be looking at in future to work on, if you can share that?
Sure. I have a couple projects, but Allison, you want to go first?
Sure. So Vitis Underground, this was a 5-year project that actually officially was scheduled to end at the end of August 2021, and we're going into what they call a no-cost extension for a year, perhaps more, as we continue to collect data and analyze the data that we have. Generally, what we're seeing is that the root system has a very subtle but pretty ubiquitous effect on every trait that we've measured to date in the scion. And so the question that is in our minds, this is again a team of several people at different institutions, is what can a single genetic individual do? What is the range of phenotypic variation that we can see from a single genome, and how does that change over time and over space, and even with a different root system? And another part of that is then, can those changes persist from 1 year to the next? So does a plant actually, we know that this can happen from other types of studies, learn? Does it learn in the frost example, like, oh, if I put my leaves out too early, they're going to get nipped off at the end of May. Maybe that shouldn't happen the next year. And that is the types of, I don't have an answer to that. And to be honest, I'm not sure quite how that hypothesis would be tested, but that's the kind of thing I'm thinking about, that we're just beginning to glimpse the range of traits that a single individual plant can present. So I would anticipate building that out a bit in the future, but it's all quite vague at this point.
But so interesting, when they used to think that, like, no, you don't really have that learning ability, and then there have been a lot of reports about learning and communication, and so it's super interesting field of study, right? That edge right there is really interesting.
I totally agree, and it sounds a little crazy as I said it.
I know, it's embarrassing to admit that you believe, yeah.
But that's what I'm thinking about, and so whether we can develop that into a viable project remains to be seen. But
yeah, because it's really about how fast the adaptations can occur, right? And at the individual level as well as like at the species level. So
and it's just really important for a clonally propagated crop where you have individuals that are replicated across counties and states even, how that works, so.
But that's also interesting because when I was working with clonally propagated crops with the Seed Bank project, which is still, I haven't been working with clones recently, but the project's ongoing. But that part of it, like just looking at it's a clone, but everyone is different. Right?
It's,
you know, like what those little plantlets look like. It's not a Xerox copy, or a photographic print where, you know, there's
but genetically
variation.
they're the same.
Right.
So what is it? And there's a lot of
Yes.
work in that area. I agree that is the edge, and it's super interesting.
Yeah, that is super interesting.
Yeah.
So what I'm working on is I started a couple projects recently. One is, I can't really talk about it, but the second one is I'm working with the Botanical Research Institute of Texas. And I'm working with a scientist named Alejandra Blasco, who's working on the flora of Colombia. And then I'm also working with Tiana Raymond in her in the herbarium. And what we're looking at, again, super at the beginning, I'm working through their archives and looking at the way that people that make objects to understand and do research on botanical specimens. So I found a cache of these very, very interesting clearings, which I'm sure you work with all the time, that are from a researcher that was working in the mid-century. And those have led me to all these other questions about how science is done now, how they're studying plants now, what's still interesting, what is still useful, right? You know, there was a brief moment where before DNA research came in that people were talking about, well, maybe all we need in a botanical library is the picture, right? Before they realized that the DNA was still in those plants, right? So I'm looking at that and really finding my way, but there's a show in 2 years. So I'm going there weekly, and it's just a treasure trove.
I'm sorry to say that we have to wrap up. As always, speaking with you both is so enlightening, and even though we've discussed this subject numerous times, I mean, obviously it's the first time for this audience, but the fact that you always present the information so succinctly and how you demonstrate the intersections, I think is one of the reasons that I'm so thrilled that the two of you together were able to be here this evening. So thank you so much for doing that. It's been an absolute delight and pleasure to have you here and to work with you on the Grafting the Grape exhibition. And thank you for sharing your artwork, Dornith, and your science and both of you, your passion for plants and sharing your information with the public. We're just really lucky to have had your expertise here this evening. So thank you so much.
Oh, thank you.
Thank you.
It's a pleasure.
Thanks so much to everyone who joined us this evening. Our last talk will be on September 15th, again at 6 p.m. And the other artists in the exhibition, Lehan and Lorraine Walsh, will be joining us also for a dialogue, not in the galleries, unfortunately, but they'll be speaking about their work in the show. And so I want to thank everyone who joined us this evening, and it was great to have you here. And thank you again, Allison and Dornith. And have a good evening, everyone.